Hello, everybody. Welcome back. Today we are beginning the first of two chapters that look at the plant kingdom. And what you're going to find as we work our way through the plant kingdom is that much of what you thought you knew about, particularly about sexual reproduction, you're going to have to rethink as we look at the plant life cycle, because this is the first time that we encounter a simple observation that the animal life cycle, particularly with respect to sexual reproduction, is not a good framework to measure other life cycles against because of the relationship between meiosis and sexual reproduction. And of all the things that historically I've noticed that students struggle with in this class, particularly when it comes to plants, is understanding the plant life cycle. So I'll spend a little bit of time when we get to the generalized life cycle, and also when we get to the specific life cycles that you're accountable for, two in this chapter and two in chapter 30, going over those life cycles and explaining to you how those life cycles are variations of the generalized life cycle that we'll encounter first. It is really important that you come to terms with this change in understanding of life cycles, because it will drive you a little batty if you don't come to terms with the differences in the life cycles. You'll be constantly answering questions incorrectly because you'll be constantly referencing to an animal life cycle, not to a plant life cycle. And you'll find it very frustrating, let me put it that way, if you don't try and make sense of these life cycles. So we'll spend a little bit of time talking about that. The other thing is that there's a whole slew of new terminology that you're going to have to come to grips with as well. So I'll spend some time with terminology along the way, make sure that I clearly define all the new terms that you're going to have to become familiar with. And what makes it a little bit difficult is that many of the terms sound very similar, but have very distinct, different meanings. So it makes it a little bit difficult to come to terms with the terminology, but again, just as coming to terms with the life cycle is important, so is coming to terms with the terminology and making sure that you understand the subtle differences or the big differences between some of the terms that we're going to encounter along the way as we talk about plants. So that being said, let's make a start talking about plants. So the first thing to deal with is just the idea that we are so comfortable seeing a landscape with plants. In fact, all of our ecosystems are defined by what type of plants are present. So when you think about a rainforest, you have a certain image of what a rainforest looks like, and I'll guarantee it's pretty much based on plants. If you think in terms of tundra or prairies or any type of ecosystem that you might imagine, the image that first comes to mind is the landscape with plants on it. Yet if you go back no more than about 500 million years, you would find that dry land would be barren. There would be no plants, because we don't see the first evidence of plants as terrestrial residents until approximately 470 million years ago. It may have been a little bit before then, but around 470 million years ago, we find fossils that at least have the appearance of plant tissue, letting us know that at this point in time, at least the early representatives of the plant kingdom had begun the process of colonizing dry land. So it's really hard to imagine a landscape with no plants in it. Even a modern day desert can still have plants. And it would be very unusual to find a landscape that is completely and utterly devoid of plants. And yet, for the vast majority of the history of life on Earth, that was the case. Dry land had no plants. But today, we know that plants form the foundation of every ecosystem. as I said, we name our ecosystems on the basis of what plants are there. And the reason is quite simple, because it's the type of plants that are present that determine what other species, animals, fungi, and so forth, can live in those landscapes as well. And so as we talk about the plant kingdom, we need to bear in mind that what we're really talking about is the colonization of dry land by everything that we are familiar with today. Because without the initial colonization by plants, life on Earth as we know it, on dry land at least, could not exist. So that puts them in a very important position. And coming to an understanding of how they came to colonize dry land and their role in the ecosystem becomes incredibly important. So let's move on from there and take a look at where we think they came from. We know that there is a lot of shared biochemistry between plants as we know them in the plant kingdom and algae. So that suggests very strongly that the ancestors of the modern plant kingdom must be found amongst the broad array of algae that we find either extant today or present in the fossil record. When we look at that huge diversity of algae, of course, we focus on green algae because plants are green, the presence of chlorophyll A and B being very distinctive as their photosynthetic pigments. And we do know that there are other connections to algae in terms of the biochemistry and the genetics of photosynthesis. But while there is a large variety of green algae, there should be one line of green algae that have the closest relationship to all of the plant kingdom. And when we try to figure that one out, a group of algae known as charophytes, or charophyceans, sometimes referred to as the charophyta, stand out as being the potential origin of the plant kingdom. And there are a number of reasons for this that we can point to, but there are 4 distinctive reasons that we can point to. The first is that cellulose, as the primary polymer found in plant cell walls, is excreted from a complex called the roseate complex. When you look at it embedded in its embedded location in the cell wall of plants, the cell membrane of plant cells, you see that it's made up of a cluster of proteins that are embedded in the membrane with a hollow tube in the center from which the cellulose fibers are excreted. And while there are a variety of these complexes found across algae, it is only amongst the charophyceans that we find the actual roseate complex that is shared by all members of the plant kingdom. So immediately we start to think in terms of the charophyceans as potential ancestors of all plants, because here is a primitive trait that's shared between all members of the clade kingdom Plantae and these algae, which would suggest that there is a connection to them. Then when we take a look at the peroxisomes, and plants are loaded with peroxisomes because they have to deal with all sorts of oxygen-free radicals. After all, oxygen is a primary product of photosynthesis, so it stands to reason that there's going to be a lot of oxygen-free radicals around. You find that the peroxisomes contain enzymes to deal with these oxygen-free radicals. And when we look at the suite of peroxisome enzymes that are located in all members of the plant kingdom, again, it's the peroxisomes of charophyceans that contain the same suite of enzymes, suggesting again that the charophyceans represent the line of algae most closely related to the kingdom Plantae, all members of the modern kingdom Plantae. Then when we look at the structure of the sperm, now here we're thinking in terms of the flagellum as we find it in eukaryotes. Remember, this is an extension of the cytoskeleton. It's got the microtubules running up the center of it, representing the supporting structure of the flagellum, and it is completely surrounded by cell membrane. So it is an actual extension of the cell proper. When we look at the basic structure of the sperm in the Charophyceans, and then we look at the structure of the sperm in all members of the plant kingdom that also share a flagellated sperm, which are going to be the members of the plant kingdom that we look at today in this presentation, their sperm have essentially identical structures internally. And so that again strongly suggests that the Charophyceans are the algae that are most closely related to land plants. The other members of the green algae, the chlorophyta, that do have flagellated sperm, which is a large number of them, do have a very different structure to their sperm that is very different from the Charophycean / plant sperm. So again, it differentiates the Charophyceans as the most likely algal ancestor of the plant kingdom. The final characteristic that is shared by plants and Charophyceans is the structure that forms when the plant cells or algal cells undergo mitosis. So when the cells are copying themselves. You should recall from your Biology 1 course that a cell plate forms along the equatorial plane of the dividing plant cell, and that forms out of vesicles that are enriched in the cellulose secreting structures, and that those vesicles fuse together to form this elongated structure called a cell plate, which ultimately elongates and extends in two dimensions. I was almost going to say 3 dimensions, but it extends in 2 dimensions until it reaches out and merges with the surrounding cell wall of the original parental cell, thus completing cytokinesis by dividing the original parent's plant cell into 2 daughter plant cells. The formation of that structure, known as a phragmoplast, and the supporting structure that guides the formation of the cell plate, again referred to as a phragmoplast, is uniquely found in all members of the plant kingdom and in the Charophyceans, nowhere else amongst the algae. So these 4 structural features strongly link the Charophyceans to kingdom Plantae as the closest living relatives. Then we take a look at the chloroplast genetics and the genetics or the genes that have been transferred through horizontal gene transfer from the cyanobacterial ancestor of chloroplast into the host cell's nucleus, because just like mitochondria, there has been horizontal gene transfer from the bacterial, in this case cyanobacterial ancestor of the chloroplast, into the host's genome, the nuclear DNA. When we look at the genes that are of interest that are associated with photosynthesis, we find again that the genes, particularly those that are associated with photosynthesis in the chloroplasts and in the Charophyceans, are extremely highly conserved in terms of their gene sequence, which tells us again, or suggests very strongly again, that the Charophyceans really are the group of algae from which would emerge the earliest members or the earliest terrestrial living ancestors of what would become the plant kingdom. So for these reasons, we point to the Charophyceans as the ancestors of land plants. That Charophycean ancestor is long since extinct, of course, but we do have around 30 species of Charophyceans that are extant today, and they all share a common feature that we think probably also makes them the prime candidates for the ancestors of modern land plants. And that is that they all grow along the margins of bodies of fresh water, whether they be streams or freshwater lakes. And in growing in those regions, they would be subject to falling and rising water levels, meaning at times they would be immersed in water, but at other times they would be exposed to dry land. And if this was in fact the environment that the early Charophyceans were growing in, those that would give rise to the plant kingdom, it naturally stands to reason that natural selection would strongly favor those Charophyceans that were able to tolerate exposure to dry land, initially for short exposures, but progressively longer exposures as the environment changes and the water levels rise and fall. And if you get an accumulation of traits that allow for a greater tolerance of exposure to air and to dry land, eventually one would expect that you would accumulate a significant enough level of traits to allow you to permanently colonize dry land and not have to be periodically immersed in water again. And these would become the ancestors of land plants as we know them today. So here are a couple of members of the modern clade Charophycia or Charophyta. So there's Chara itself, which you've already seen when we looked at protists, and another type of Charophyte that lives in ponds and particularly along the margins of freshwater ponds. So once we recognize these as being the ancestors of the plant kingdom, then we have to ask ourselves, how are we going to define the plant kingdom? There have been a number of attempts at trying to create a broader definition of the plant kingdom. The textbook uses the traditional definition that members of the kingdom Plantae are embryophytes. And what that means is that these are organisms, plants, that produce embryos as part of the reproductive process. And when we look at the life cycles, I'll show you where the embryos exist within the life cycle. But this is not the only way in which the plant kingdom has been defined at various points in time. And so a couple of other definitions you can see illustrated here on this diagram. So here's Plantae as embryophytes and strictly as embryophytes. But given that charophytes are so closely related to terrestrial plants or to the kingdom Plantae, several people, well, a number of people, let's put it that way, have suggested that maybe we ought to create a clade that includes the charophytes, given that they're so closely related. And this clade has been tentatively labeled Streptophyta. So you may find this referenced in some of the extracurricular study that you may do outside of the textbook. If you take a look at some of the videos that I've posted in the resource section, or if you take a look at other material that you might find on the internet, you may see reference to Streptophyta. This is simply an attempt to include the charophytes as part of a broader photosynthetic group that includes both the terrestrial plants and their closest living algal relatives. Some people have gone one step further and said, given that green algae in total, the chlorophytes, also share a lot of similarity with terrestrial plants in terms of photosynthesis and chloroplast, perhaps we ought to create a clade that incorporates all those that trace their chloroplasts back to the primary endosymbiosis that gave rise to the original chloroplast, which was almost certainly a green algae, because the cyanobacteria themselves have the chlorophyll A and B and are green in color as a consequence of that. And so, again, a tentative clade called Viridiplantae, which, if you want to translate it from the Latin, means green plants, because viridi comes from the root Latin word for green. So you basically got green plants, which refers to anything that traces its origin back to the original, as I said, endosymbiosis that captured the cyanobacterium and gave rise to the original green-colored plastid because of the presence of chlorophyll A and B as the primary photosynthetic pigments. There is still an ongoing discussion as to whether we should expand the traditional plant kingdom, the embryophyte-based plant kingdom, to include the charophytes or to include all chlorophytes. But given that the textbook is using just the embryophyte definition, that's what we'll follow in this presentation as well. That will reduce some of the confusion that you may encounter or may feel as we work our way through the diversity of the plant kingdom. Now, there are a couple of other terms that are associated with this notion of embryophyte that I also want to introduce you to at this point. By virtue of the fact that it has relevance to the life cycle, and we're actually going to encounter one of these terms very shortly as we look at the derived traits that define the plant kingdom. So the two terms that I want to bring up are sporophyte and gametophyte. And in fact, you're going to find that these terms are going to define the two major time periods in the plant life cycle. We actually divide the plant life cycle into two major periods, one known as the sporophyte generation and the other known as the gametophyte generation. And you will have encountered this already in the Plants 1 lab that you've just completed. But here, because we want to dig more deeply into an understanding of the plant kingdom, you're going to have to really come to terms with these two terms. So sporophyte refers to the diploid stage It's the diploid stage of the life cycle, and it produces spores. And they are actually the product of meiosis. And this is where we start getting into one of those observations of the life cycle. You have probably always considered that the cells that are the product of meiosis, the haploid cells that are the product of meiosis, those are going to be the gametes. And that would be true if you were thinking animals. But when you start thinking plants and when you start thinking fungi, you have to drop that idea. The product of meiosis is a haploid cell known as a spore, not a gamete. The gametes are produced by the gametophyte. And here's where the surprise comes. Because the parent cells in the gametophyte are haploid, the gametes are actually produced not by meiosis, as you might traditionally think, but by mitosis, because gametes are still haploid. And if you're starting with a haploid cell, to give rise to a haploid cell, you don't need to use meiosis, you need to use mitosis, because remember, mitosis makes identical copies of the parent cell. So if you're starting with a haploid cell and you want to produce a haploid cell, mitosis is the way you go. So gametes in the life cycle of plants, and as you'll see in a couple of weeks in fungi, are the product of mitosis, not the product of meiosis. So this is going to become one of those things as you start to pay attention to the life cycles. And you will be responsible for knowing 4 life cycles and being able to draw them out and be able to place the various stages into the life cycle and identify where diploid and haploid is concerned and how the gametes are produced and where they're produced and what the reproductive structures are. So you're going to have to dig deeply into the life cycles. it's really important that you understand this difference between these two stages and recognize what this means, because it has implications. So why introduce it here? Well, remember that when we talked about embryophytes, an embryophyte produces embryos. A sporophyte produces spores. A gametophyte produces gametes. So these are the reasons why, or this is the reason why I wanted to introduce these terms. Now, plus it gives me now an opportunity to use these as we talk about some of the derived traits that identify plants as a clade and separates them from the charophytes. So the first trait that, or another trait that is shared with the charophysians, and remember again, we think the charophysians that give rise to the plant kingdom probably live in similar sorts of habit, or lived in similar sorts of habitats as the extant charophysians, that is along the margins of freshwater bodies, where the water level is constantly going up and down seasonally. That these charophysians probably also contained this polymer called spiropolymin, that in the extant charophysians and also in members of the plant kingdom form a wall that surrounds the zygotes, what we might refer to as an environmentally resistant coating around the spores to prevent them from drying out, because the spores are what are typically released by many members of the plant kingdom into the environment. And those spores have to germinate and are going to develop into a structure known as the gametophyte, a multicellular haploid structure. So being able to protect the zygotes from drying out until such times as they encounter suitable conditions for them to germinate and begin cell division, it's important to have an environmentally resistant coat. So the presence of spiropolymin is one of those things that we know is again another primitive trait that was inherited from the charophysians, and the modern charophysians certainly share that with members of the plant kingdom. But there are 4 traits that are considered to be diagnostic in terms of derived traits that identify members of the plant kingdom as members of the plant kingdom and separate them from the charophysians. So here are, I said 4, I've split them up into 5. Okay, so we'll take them as 5, and the reason why is because having already talked about the walled spores, I've got that included in here. That's connected with the spiropolymin. So I've listed them as 5 traits. So the first is something that may come as a surprise to you. If you've been paying attention to the algae from chapter 28, where we talked about algae as members of the protists, you may recall that the life cycle of the vast majority of algae that we identify as protists, whether they're unicellular or multicellular, show an alternation of generations, particularly in the multicellular ones, the ones that we would refer to as seaweeds. You have a multicellular diploid generation and a multicellular haploid generation, and the life cycle cycles between these 2 generations of multicellular forms. What is rather surprising, and what makes this a derived trait, is that of all the algae that are multicellular, the charophysians do not show alternation of generations. They are the one form of multicellular algae that do not show alternation of generations. So given that we point to the charophysians as the ancestral group of algae that give rise to land plants, to not have alternation of generations in the charophysians, but to see it amongst all representatives of the plant kingdom, means that alternation of generations, as we see it in the plant kingdom, arose after the earliest members of the plant kingdom diverged from the common ancestor with the Charophysians. In other words, it's a derived trait. It's not a shared primitive trait with the Charophysians. This is a shared derived trait amongst members of the plant kingdom, not shared with the Charophysians. The second relates to this definition of the plants as being embryophytes. The fact that they produce a multicellular embryo that is supported by the parental plant for some portion of its early development until such times as it reaches a point where it can survive independently, or potentially survive independently, depending on the type of plants that we're talking about. More of that to come. Then, of course, we have the presence of the sporopollenin forming a walled structure around the spores that are produced in a structure called the sporangia, which is found in the sporophyte. So now we start to come across some terminology that we need to pay attention to. I was about to write a definition here for sporangia, but I know I've got slides coming up where we look at this notion of sporangia in a little bit more detail. So I'll save the definition for there. Then I've talked about the notion of a gametophyte as a multicellular haploid structure in a previous slide where I introduced you to the term. Within the gametophyte, there's got to be a structure that gives rise to the gametes, and that's known as a gametangium, plural gametangia. You can see the term here. And that structure is going to be multicellular. So again, this is a structure that we do not find in the Charophysians. We do not find it amongst the algae as a general reproductive structure, but it is found pretty much universally across all plants until we get to seeded plants, where we don't have a gametangium as a formal structure, but we do have a multicellular haploid structure. We just don't refer to it as a gametangium. And then something that is really unique about plants that sets them aside from all other organisms is the presence of apical meristems. This is a piece of tissue located at the tips of every root and every shoot, where cell division continues throughout the life of the plant. This is the point from which those structures grow in plants. So plants grow from the tips of the shoots and tips of the roots. They don't grow from the base of the stem. One of the questions that I used to frequently ask my plant physiology students when I taught plant physiology over at UCF was, if you were to drive a metal spike into the trunk of a tree 6 feet from the ground and come back 50 years later, where would you expect to find that spike? If they remembered that the trees were growing from the meristems at the tip of the shoot, that is at the top of the tree, then they would answer approximately 6 feet from the ground. If they forgot that, then they would say the spike would be somewhere high up on the trunk after 50 years. And again, this is a misunderstanding of where plants grow from. They grow from the tip of the shoot and the tip of the root. They do not grow from the base of the stem. So that's very important to remember. So let's take a quick look at each of these so that you can see a little bit more detail. So the alternation of generations is this idea of going between two multicellular stages, one being haploid, one being diploid. We have to remember that the product of meiosis is not the gametes, it's the spores produced by the sporophyte. So meiosis, meiosis gives rise to spores. Okay? Meiosis gives rise to spores. And that takes place in the sporophyte, which is itself diploid. The gametophyte is the product of the germination of the spores. So they develop into a multicellular haploid structure. And that's the gametophyte. And that gives rise to gametes. So a sporophyte produces spores by meiosis. The gametophyte produces gametes by mitosis. So again, haploid gametes are produced by mitosis in the gametophyte. Okay, so this becomes really important for you to understand. This is going to be a source of much frustration as you try and understand the life cycle of plants, unless you wrap your head around this idea. You have to discard what you learned about animals. You cannot compare this to animals. You cannot use the animal model. And I'll keep repeating that multiple times. And if you start struggling and you come to talk to me about this, I'm going to ask you about the life cycle and how do you understand the life cycle? What is the product of meiosis? Where do gametes come from? Where do spores come from? What does alternation of generations mean? Because it's really important that you come to terms with this. It is something that we will repeat again when we talk about fungi. You're going to see the same basic idea. And you, again, will feel very frustrated if you don't come to terms with this idea when you're thinking in terms of the life cycle. So here is the generalized life cycle. And so here is the sporophyte, which for most plants is what you're going to see in the environment. So when you look at a fern, when you look at a flower, when you look at a tree, you're looking at the sporophyte. There is one group of plants where that is not true, and we'll talk about those a little bit later in this presentation because it comes up there. So the sporophyte has a sporangium which is where meiosis occurs to produce the spore. So the sporophyte, somewhere in the sporophyte, you're going to find the sporangium. The sporangium is where meiosis occurs, and that gives rise to the spores. The spores then germinate to produce the gametophyte. In the gametophyte, there is a gametangium. That's not very neat. Take that out. So the plural of sporangium is sporangia. The plural of gametangium is gametangia. And depending on the type of plants we're talking about, there could be a male and a female gametangium that are located on separate individuals, or could be located on the same individual, depending on what type of plants we're talking about. So the gametangium is found in the gametophyte, and it's the gametangium that produces the gametes by mitosis. So you will get an egg, you will get a sperm, or you'll get the equivalent of an egg and a sperm, again, depending on the type of plants we're talking about. Those gametes then undergo fertilization, and when they come together, that gives rise to the zygote, which then, by mitosis again, develops into the sporophyte. So when we look at the life cycles that we are going to be looking at in this chapter and in the following chapter, I'll point out to you which part represents the gametophyte, which part represents the sporophyte. General rule of thumb, which you may not have picked up on from the lab, but this is something that you probably ought to know. So gametophyte generation is haploid. So anything that is haploid is considered to be part of the gametophyte generation. So that means that spores, even though they are the product of the sporangium and the sporophyte, are considered to be part of the gametophyte generation because they are haploid. The sporophyte generation is diploid. So anything in the life cycle that is diploid is part of the sporophyte generation. So the zygote is itself part of the sporophyte generation. And again, you can see how that's been divided out here in the life cycle. So anything that is blue is the sporophyte generation. Anything that is red is the gametophyte. It is the sporophyte generation. So this is gametophyte. And this is sporophyte. So hopefully you can master that relatively quickly because it's an important idea to master. Let's move on here. So multicellular dependent embryos. This is something that all land plants produce. In fact, let me talk very quickly about something that we'll encounter next week when we talk about seed plants. But when you split a seed open and you take inside a seed, you're going to see a tiny little body somewhere inside the seed. That is in fact the little embryonic plant that is being packaged inside the seed for distribution. That is the advantage of seed plants, that they're not releasing spores into the environment. They're actually releasing a multicellular embryonic plant into the environment via the seed, packaged with nutritional material, because that's what the large part of the seed actually is. And so here you see embraced in seed plants this notion of an embryophyte producing embryos. That embryo actually develops within the body of usually the female structure within the plant if it contains both male and female, or in the female plant if there are separate male and female plants, which is not unusual in the plant kingdom. And these embryos during their initial development are fully dependent upon the parental plant, and they get their nutrition through a group of cells, which you can see referred to here as placental cells, because they do essentially the same role as the placenta does for placental mammals. It allows for the transfer of nutrients from the parent plant into the developing embryo to support its growth. So here you can see the developing embryo. So here's the embryo inside Marcantia, which is a liverwort. And liverworts are considered to be I remember now. Liverworts are actually considered to be the earliest, okay? Yeah, in terms of where we currently place them, they are considered to be the earliest or the most basal of all members of the current plant kingdom to the plant taxonomic tree. So you can see that even in the what are considered to be the most basal representatives of the plant kingdom, there is an embryo. This is actually inside. So this would be inside the female gametangium. Because in liverworts, the egg develops in the female gametangium, which is called an archegonium. I'll put that in here too. And maybe I should put here male. Male gametangium is called an antheridium. So typically the egg develops inside the archegonium and is held in the archegonium. The sperm being flagellated in the plants that we're talking about today can then swim from the antheridium, the male gametangium, to the archegonium, the female gametangium, for the purposes of fertilization. The zygote then forms in the gametangium. So this develops from the zygote. So the fertilization event takes place within the female gametangium, the archegonium, and the subsequent zygote that forms as a consequence of that then starts cell division and develops into the embryo inside the gametangium. Ultimately, it will then grow out of the gametangium, still being supported by the female gametophyte via the gametangium. This is something that is unique to the life cycle of liverworts and their relatives, the mosses and the hornworts, what are known as bryophytes. More of that to come. Then we've talked about the fact that there are walled spores that contain the sporopollenin to prevent them from drying out. So within the sporangium, so here is the plural term, sporangia, in the sporophyte, there are sporocytes. These are the cells that undergo meiosis to produce the spores. And those spores have the sporopollenin to protect the spores and prevent them from drying out. So if we look at the slide, here is the sporangium in which the sporocytes are located, and those sporocytes are going to produce the spores by meiosis, and those spores will ultimately be released to settle in the environment and hopefully find useful or suitable conditions so that they can germinate to give rise to the next generation of plants. This is in a moss. Otherwise, this is sphagnum moss, and you're going to see that this is fairly characteristic. So you've got the gametophyte on which the sporophyte is actually growing. So So a distinctive part of the bryophyte life cycle is the fact that what you see in the landscape is not the sporophyte, but the gametophyte. The sporophyte grows out of and is dependent upon the gametophyte, and it's transitory. It only exists long enough to produce the spores and release those into the environment. Those spores then germinate to give rise to the next generation of plants, but the next generation of plants emerges as gametophytes. They are the dominant form, and this is unique to the bryophytes. It's what sets their life cycle apart from the life cycle of all other members of the plant kingdom. And that is a reflection of the fact that they are most basal to the plant kingdom and that they are sharing some characteristics in terms of their life cycle with that of the algal ancestors, because remember, in many algae, the dominant form is the haploid form. So here we see that the haploid form is still the dominant form after starting to colonize land. This would change. The relationship between the sporophyte and the gametophyte is actually one of those distinctive features that we recognize as we see the evolution of the plant kingdom. And we'll talk about some of those changes. We'll see one of them Today we'll talk about some more in the next chapter when we do chapter 30. Then we have the multicellular gammatangium. I've introduced you to these terms. So we have the archegonia, that's plural. Archegonium is singular, that's the female gammatangium. And then the male gammatangium is called an antheridium. So if you want to make it singular, you replace the A with a UM. So it's antheridium, gammatangium, archegonium. Those are the singular. So the female gammatangium, the archegonium, produces eggs. The male gammatangium, the antheridium, produces sperm. Remember, in the basal representatives of the plant kingdom, they have flagellated sperm. That will change. We'll see that when we do the next chapter. So here you can see the female gametophyte. This is again liverworts. Marcantia is the liverwort that we look at the most. Sphagnum moss or peat moss are the two mosses that we look at with the mosses. We don't look a lot at hornworts, but we'll look at a representative hornworts when we get to looking at the bryophytes as a group. So here you can see the archegonia and you can see the antheridia. And remember, the sperm are going to swim from here into the archegonium to fertilize the egg, and the embryo is going to develop in there, as we saw in a previous slide. So the sperm swim from the antheridia across to the archegonia and fertilize the egg. And that puts limits on the bryophytes, as we'll talk about when we get to the bryophytes. It obviously put limits on the early land plants as well. It kind of explains why they probably spent a lot of time very close to water and lived in fairly damp environments early on. Then the apical meristems. This is the tissue where cell division continues throughout the life of the plant. So its lifetime. And plants show what's called indeterminate growth. Meaning they continue to grow until genetically programmed to die. I think there should be two M's in there. Or something kills it. So they're constantly growing, constantly getting bigger until such times as they are genetically programmed to die or they get killed off, which means we have annuals, 1 year, biannuals, which is 2 years, and then perennials. And this is why we have things like the bristlecone pine and other plants that have been dated to around 4,000 years of age and still growing. There is some suggestion that the bristlecone pines, while being the oldest of the trees, may not be the oldest plants on the planet. We think that some creosote bushes growing in the Mojave Desert of Arizona and Southern California may be 12,000 years old or more. That's based on the fact that creosote bushes grow out in a ring fashion from the original bush at that started it. So they expand out in a ring fashion. The center of the ring dies out, leaving a hollow ring, and you can extrapolate backwards, knowing the rate of expansion of the ring, because it's a fairly constant expansion rate, to the point in time when the original bush grew from the germinated seed. And some of these creosote bushes in the Mojave Desert may be 12,000 years or more in age based on that extrapolation. There are some other plants that I'm aware of in Australia. There's a couple of bushes in Tasmania, the large island off the south coast of Eastern Australia, that are suspected to be upwards of 25,000 years in age. And the reason why they can grow for this length of time and get this old is simply because they have this meristem tissue that allows them to keep growing and just to keep getting bigger. And it's because of where the meristem tissue is located. So it's located at the tip of the root. So this is the root cap. So this is the sacrificial tissue on the tip, the very tip of the root, that gets scoured away as the root forces its way through the soil. Then behind it is the meristem. And here, every time a cell divides, one of the cells remains in the meristem to continue the division process. The other cell remains in this physical location. The root continues to grow in this direction. This area is going to remain static, and this tip is going to move, is going to continue to grow away from it. And that allows differentiation of those cells into whatever type of cell it will become based on its position within the root, while the meristem cells continue the growth of the root in this direction. Same thing happens in the stem. You've got the meristem right in behind the apex of the shoot. One cell remains in the meristem after each cell division. The other differentiates into part of the stem on the basis of where it's located. Now, there are two other types of meristem. There is the cambium layer. That's not as neat as I would like. So let me, oops, guess I should rub it out. There we go. So this is meristem. That produces xylem and phloem and woody plants. So this is another type of meristem that's located underneath what most people refer to as the bark. In the plant physiology world that I come from, we refer to that as the cork layer. So cork is the outer layer, and what you actually know as cork is derived from that layer on certain types of trees that produce very, very thick layers of material. And so a tree expands in girth, in diameter, as a consequence of the activity of the cambium layer. And then there's also, and you can just see it here on this diagram, this is called an auxiliary bud. So there's a little bit of meristem that's left behind here in the auxiliary bud as the shoot continues its growth up here. And the primary shoot, this area, sends out chemicals that stops this from growing. But if you take this off, so if you trim your bushes so that you cut them back, you're basically taking off these primary shoots. That removes the chemical that's stopping the auxiliary buds from growing. They now begin to grow, and that's what leads to the growth of a bush rather than a tree. And so there are plants that we have in the landscape that could either be a tree or a bush, depending on whether you trim them or not. They could develop into a hedge, or they could develop into a tree, depending on whether you keep cutting them back and taking off the primary shoots all the time. And again, it's the meristem that allows this to happen. So the primary meristem is the tip of the roots and the tip of the shoots, and that's where most of the growth in plants takes place universally. In woody plants like trees and shrubs, you have the cambium layer that allows them to lay down material and expand the girth or the diameter of the trunk, and provides new xylem and phloem, the fluid carrying tissue, every year. And then you have the auxiliary buds that can grow if the primary buds are removed. And all of this contributes to the growth of the plant and shapes the growth of the plant over time. Then plants are also really excellent organic chemists. They produce a wide range of organic molecules to protect themselves from ultraviolet radiation, to provide waterproofing. It talks about additional compounds found in the cuticle. So something that you may be familiar with, carnauba wax. So there's the carnauba tree in Brazil that actually produces carnauba wax. If you put wax on your car, it is likely a carnauba wax that was derived from these Brazilian trees. We simply take advantage of the fact that it's extremely hydrophobic. It is part of the cuticle that allows the plants to reduce water loss, because exposure to air, once you move out of the water, is going to become a big deal. And so you have to protect yourself from that. You have to protect yourself from ultraviolet radiation. You have to protect yourself from potential mutation, because remember, for early plants, the dominant generation is the haploid generation, not the diploid generation. So that makes them very susceptible to mutation. There's a whole host of issues that they have to come to terms with. The book walks you through some of those. Perhaps the most significant, of course, is being able to obtain nutrients, both in the form of water and minerals out of the soil, something which they didn't have to worry too much about while they were totally immersed in water. And what we do know is that the tissue that would ultimately give rise to roots as a mechanism for anchoring the plant to the ground and also providing access to water and minerals would not evolve until much later, perhaps 30 or 40 million years after the earliest plants began the colonization of dry ground. So one wonders at this point, and this is a question that biologists have asked, how did these early colonizers manage it on dry ground if they didn't have roots and shoots and leaves, because all of those require vascular tissue, and allow plants, the earliest plants, to colonize land? And the answer is found in the fossil record, and also found amongst the plant kingdom today. And the answer is fungi. We know that about 90%, maybe 95% of all members of the plant kingdom, modern plant kingdom at least, form some form of symbiotic relationship with fungi. And in that symbiotic relationship, the fungi are providing access for the plant to extra water resources and extra mineral resources. In return, the plant provides the fungus with access to products of photosynthesis. So this is a mutualistic, in general terms, relationship. And based on the fossil record, this interaction between plants and fungi goes back essentially to the point at which the earliest members of the plant kingdom can be identified in the fossil record, which suggests that the success of plants as colonizers of dry ground may in fact be a function of their symbiosis with fungi, and that it was in fact the fungi acting as substitute quote unquote root systems for these early plants that allowed them to successfully colonize land in the first place. Ultimately, they would evolve their own ability to be able to extract water and minerals out of the soil, but they would not discard their mutualistic relationship with fungi. That would continue. And we will investigate that a little bit more when we get to chapter 31 and we talk about kingdom fungi in more detail. So when do we see that first evidence of plants or plant tissue that we can identify specifically as plant tissue in the fossil record? Well, this is the very, very earliest plant tissue that we are confident is plant tissue, and it's sporophyte tissue. How do we know? Well, you can see some pictures here of structures that are interpreted as plant spores. Why? Because they look very similar to spores that are produced by extant plants today. Those were found embedded in this tissue. They come out of this tissue. And so this, if these are in fact plant spores, this must be sporophyte tissue. And sporophyte tissue, multicellular sporophyte tissue like this, is characteristic of plants. And this is roughly 470 million years old. Sorry, 400 and, let me go back to the previous slide, make sure I get the time. 425 million years old. So these are Chinese fossils that have been dated to, as I said, to about 425 million years old. And it is clearly sporophyte tissue because of the presence of spores, assuming that this is spore tissue. One of the earliest plants that is represented in the fossil record is this plant known as Cooksonia. And when you look at Cooksonia and you look particularly at the sporangium, with the exception of the shape of the sporangium, which is a flattened disc rather than a more elongate semi-ovoid structure, this Cooksonia looks remarkably like some modern day spore-producing plants. If I remember rightly, the comparison is being made to lycophytes, either lycophytes or some of the bryophytes. I can't remember which the comparison is being made to, unfortunately, right now. But I know that this tissue is almost, that the structure of a Cooksonia, as we see it in the fossil record, looks very remarkably like the sporophytes of some extant plants. So we're pretty confident that Cooksonia represents the sporophyte, the Cooksonia fossils that we have represent a sporophyte of a plant, but exactly where in the life cycle this occurs is not entirely clear. So it may be bryophyte, it may be lycophyte. We'll be looking at both of those today. What that also suggests is that if we've got this sporophyte tissue dating from this point in time, then it suggests that the earliest ancestors of the modern plant kingdom to begin the colonization process probably did so sometime substantially earlier. So the date for that is usually pinned at around 470 million years ago. Give or take a little bit of flex time, but usually around 470 million years ago is around the time that we think the earliest plant ancestors began the colonization process. And they almost certainly did so accompanied by fungi to allow them to extract water and minerals from the soil. So this is what we now think is the phylogenetic tree of the modern plant kingdom. So liverworts are most basal, but we think they share a lot of characteristics with those very earliest common ancestors of all the plant kingdom. Then the mosses are considered to be the next most basal, which is somewhat ironic given that mosses do show the presence of very in the case of some mosses, some very primitive vascular tissue. And also you may recall from the phylogeny lab, the taxonomy lab, that some mosses also possess stomata, something that you're not going to see until you see the evolution of leaves. And so somewhat ironically, mosses are considered to be a little bit more distantly related to all vascular plants than the hornworts. Hornworts are considered to be the most closely related to vascular plants. The actual origin of vascular tissue is somewhat murky. We don't have really good fossils to illustrate how vascular tissue may have emerged, but there are some ideas, and I'll briefly talk about those when we talk about the emergence of vascular plants a little bit later today. Then we have the lycophytes as the first group of vascular plants to emerge. Then the manilophytes, the ferns, the horsetails, and whisk ferns, which used to be once considered to be three different clades, but are now, on the basis of genetic evidence, considered to be a single clade. But as you may do some work outside of the textbook, you may also find reference to three different clades. I will give you the names of the three different clades when we get there, just so that you're aware of the fact that these are sometimes used by some taxonomists, but are not necessarily universally accepted. And then in the next chapter, we will get on to the gymnosperms and the angiosperms, the seeded vascular plants. And we'll talk about the origin of seeds when we get to those. So in terms of the groupings of plants, you've heard me talk about a number of different type of plants. So here are the informal groupings. And what I want to point out, and something that gets very confusing again for Bio 2 students who first hear this, you've got this term bryophyte, but then you've got bryophyta, which are the mosses. Anthocerophyta are the hornworts, and hepatophyta are the liverworts. So bryophyta is a taxonomic term. Bryophyte is not taxonomic. And it's really important to remember that. So bryophyte is not a taxonomic term. Bryophyta is a taxonomic term. So we need to make that distinction. So the bryophytes are non-vascular plants. These are the most basal to all of the plant kingdom. Then we have the seedless vascular plants, represented by the lycophyta, sometimes also referred to as lycopodophyta, depending on who's doing the taxonomy. Then we have the manilophyta. These are the ferns, horsetails, and whisk ferns. These are the club mosses, spike mosses, and quillworts. And it's unfortunate that these common names have been associated with these plants because they are neither mosses nor worts. Those terms apply to the bryophytes. But unfortunately, these names were given before it was recognized that there was a distinction here. These are very old terminologies, and no one has decided to give them more distinctive common names because these names are stuck or have stuck. Then we have the gymnosperms. And the term gymnosperm means naked seed, which is very descriptive of what it's like. The seed is not protected other than by a seed coat. So you have the ginkgophyta, the cycadophyta, the gonidophyta, and coniferophyta. And then you have the angiosperms, otherwise known as the anthophyta. And angiosperm means covered seed. So the seed is inside a structure called the ovary, as opposed to the ovule, which is what you find in the gymnosperm. So these are the informal groupings, and we will follow these informal groupings. Today, we're going to talk about the bryophytes and the seedless vascular plants. In the next chapter, we'll talk about the vascular plants, the gymnosperms, and the angiosperms. So this table comes straight out of the chapter, just gives you a breakdown of the number of species in each of those groups. You can take a look at that for yourself. So we've been going for a while. Let's take a break. This, by the way, I promise you, is the longest section. The two remaining sections covering the bryophytes and the seedless vascular plants are going to be much shorter. But obviously, there's a lot of detail that needs to be given here as a lead into the plant kingdom. So we are going to take a very real break. I'm going to shut down the recording after I read out the concept check questions, and I'll see you on the other side of the break with the answers. So why are charophytes thought to be the group of algae most closely related to plants? And identify and briefly explain 4 derived traits of plants that contribute to life on land. I'll see you on the other side of the break with the answers.